Hello everyone, I'm Simone Mattei, and at EMBL Heidelberg I have a dual role. This is why I have a double affiliation. I'm team leader, and that means I have a research group affiliated to the structural and computational biology unit. I mainly work on infectious biology programs and projects and protein translation. And I also lead the service team at the Imaging Center, which I will tell you much more about. And after this morning presentations, I actually realized that I'm a product of both EMBL and EMBO, because I was an EMBL PhD student. So I was in the program a few years ago. Then I did my postdoctoral work at ETH in Switzerland while being supported by an EMBO postdoctoral fellowship. And now I'm back as team leader there. And today, my role is to introduce to you the Imaging Center and show the activities of my group and my colleagues in the transversal team infection biology. But let's start with a very general introduction. I think we can all agree that in the past 2 decades, imaging technologies are really revolutionizing how we can study life. There are some great examples of how disruptive technology has allowed us to study living organisms from their entirety up to the at the atomic level. So we have now X-ray imaging that allows us to image large volumes, intravital microscopy, mainly light sheet microscopy that was developed at EMBL, and super resolution light microscopy and cryo-electron microscopy, which were both awarded Nobel Prizes because of their tremendous impact on our ability to study molecular processes, both in situ and in vitro. So all those technologies allow us to image the life at the different scales, from the full organisms down to their organs and tissues, then to go at the cellular level, and then study at the atomic level macromolecular machineries to understand how they're structured and how they work. And this, of course, requires a lot of expertise and access to high-tech instrumentation. And EMBL research groups and core facilities are between the leaders on these fields. However, there was such a tremendous amount of requests in terms of access to the microscopes, access to the expertise, and therefore collaboration that couldn't be sustained by the research groups or the internal core facilities, which are mainly meant to support EMBL scientists in addition to scientists from the member states. And that pushed EMBL in 2016 or so to initiate a new exciting project, the EMBL Imaging Center. The Imaging Center is a brand new facility, which the main mission to provide open access to the cutting-edge imaging technologies, both in light and electron microscopy. And it is aimed at servicing the external scientific community. So the Imaging Center is truly being built for you. And we not only have all the high-end equipment available for you, but we support the users with tailored project support. Our staff is just a group of amazing scientists, all at the postdoctoral level, that help you throughout your workflows. And other core missions are, of course, as anything we do at EMBL, training. Training of the users, because of course you come and use the technology, but especially important for us is the trainer program. So training facility managers from different countries that will go back and will have a major impact in training the local community once back at home. And finally, technology development. Technology development is always at the core of our activities, and we do that in collaboration with our academic and industry partners, because our aim is to then distribute whatever we develop in a timely manner. And again, the overarching theme of the Imaging Center is really to enable the external researchers to image their biological processes from molecule to full organisms. And we are now, we've been open almost since 2 years now, since autumn 2021, for calls for any external project. So what do we offer? I mean, today, in the interest of time, I will just be able to give you a bit of a glance of what we do. Of course, in the breakout rooms later on, I'm happy to answer any question you might have. But mainly, we offer three categories of services: light microscopy, electron microscopy, and maybe even more importantly, the correlative approaches, CLEM, correlative light and electron microscopy, where the two techniques are joined to study both function with light microscopy, specificity, and structure information thanks to EM. My colleague Timo Zimmermann leads the light microscopy team, and they have really a very broad portfolio of imaging technologies at your disposal to study your questions. But I think what is really astonishing is their capability of supporting super resolution microscopy. Nowadays, thanks to the mean flux technology, we are able with light microscopy to reach spatial resolutions in the range of 1.5 to 2 nanometers. There are now published papers done with these technologies where you can observe within a living cell the steps of the kinesin motor walking along the microtubule. So this is really changing entirely what we can do with light microscopy. And CLEM, of course, combines light and electron microscopy, however, facing the challenges that this has to be done at cryogenic temperature for structural biology approaches. Therefore, the light microscopy approaches we can use are somewhat limited by the energy we can apply to the specimen without melting it. So for cryo-electron microscopy, your specimen is vitrified. It has to be kept at liquid nitrogen temperature throughout the process. So at the moment, we can use wide field and confocal imaging, but we are actively developing super resolution approaches that would be compatible with cryogenic temperatures. So with CLEM, we are again able to use fluorescence microscopy to identify the region of interest within a cell. For example, you might be interested in a specific organelle, lipid droplets, and target that for structural biology. In terms of electron microscopy, we support the users with sample preparation, so the vitrification, the rapid freezing of your specimens, the screening of the samples we've prepared. If you are working with cellular specimen, the thinning by focused ion beam milling, which allows to make this very thin 200 nanometer lamellae of cellular environment, which can then be suitable for transmission electron microscopy. So high-end data collection, both for purified specimens, if you're a structural biologist that has managed to purify a specific macromolecular complex, or high-end data collection for tomography approaches directly within the cell. If you're then interested in having a much bigger picture of what is going on in your cell, well, then the technology has to move to volume imaging, so using scanning electron microscopy for the visualization of maybe a full cell. And as I said, all those workflows are based on cutting-edge instrumentations. We have the latest transmission microscopes, both for screening and high-end data collection of single particle or tomography approaches. We have dual beam machines, the Achelos and cross beams, that can both prepare your specimen, so by fib milling to prepare the cellular lamellae, but also do volume imaging if that's what you're interested in. And those microscopes that link the two technologies. So the Eriscan is a confocal with the Eriscan detector, which is able to operate at cryogenic temperatures, so do light microscopy on a vitrified specimen. But again, access to technology is one thing, but maybe even more important is access to the expertise on how to prepare your experiments, how to prepare your specimens, and then how to best use the instruments for data collection, and finally, how to interpret that. So we really try to support the users from in any area of the workflow they might need, and if you need support throughout the workflows, we can do that. we have many examples after 2 years of operations, but I just want to bring to your attention one, because I personally think it was, it was great to see what imaging services can do for the community. In this case, we were supporting the lab of Professor John Atkins in Ireland, University of Cork, and John had a brilliant PhD student, Kate, which was able to purify bacterial ribosomes in a very specific translation state. However, they had no expertise nor access to cryo-EM devices and technologies. So they came to us with just purified specimens, and our specialists prepared the sample, screened them to identify the best conditions for cryo-EM grid preparation, then acquired a large amount of data that resulted in the determination of the ribosome map at 1.5 angstrom resolution. And just to let you understand, I mean, we had no real experience with the ribosome before, but still through just application of best practices, we were able to provide the highest resolution map available to date to a user that actually have never done cryo-EM. So this is, I think, just showing the impact that access to facilities like the Imaging Center can have on your research. Just to give you an overview of what we've been doing, you see that we are still in the ramp-up phase. In 2022, we were able to support almost 150 projects with more than 450 days of microscopies provided to users that come mainly from Europe, mainly from our member states, but actually we support also people from all over the world, India and US. We also not only serve academic users, but many industry researchers are accessing our services for cryo-EM. It's mainly pharma companies, and we do a lot of training, and that includes also job shadowing to train other facility managers and transfer the knowledge. Of course, training of individuals has an impact, but to broaden our to be more effective, we also organize symposia and courses to disseminate knowledge. So, how to access our services? Well, we are just one email further from you, so you can just drop us an email and ask for help in understanding how your project can be supported. And there are many opportunities through the research infrastructure, so the ERIC, like Instruct or Eurobiomedicine, and other European Commission funded projects that can support sometimes even entirely your project. So they might support travel, accommodation, and even all the imaging sessions that you require. So just contact us, we are there for you. So this is service provision, but we want to provide the latest technologies to you, but at the same time we don't stop there. We always push the technology development. And technology development is intertwined with biological questions. So we develop new instruments, new workflows to answer biological questions that cannot be answered with what we have right now. And this goes also into service provision, not only because whatever we develop will be transferred as a service to the community, but also the projects that comes through service provision pushes us to aim at specific technology development that are needed by the community. And we really aim to minimize the gap that exists between a new development that might happen at EMBL or somewhere else and the scientific impact, because when a new technology is developed, this has to be acquired by a company that will make a commercial product that then will be on the market. You need to acquire grant funding, installing, learning how to use it. And often, by the time you have the technology up and running in your institute, you are already behind because 10 years pass by and we are already on to something new. So the Imaging Center really aims at providing you access to the latest and greatest as soon as they are robust enough to support external users. And our technology development is really done in collaboration with our also industry partners. We have the founders partner like Zeiss and Thermo Fisher, and then we added other new partners like Aberior and Cryosol that really help us to develop at all levels, from sample preparation to imaging. And they actively work on site with us, which is part of this open innovation initiative where their research engineers and developers are on site with us at the imaging center to develop the new workflows. So we really support all sorts of microscopies, from looking at full organisms from the ocean to high-resolution structural determinations, as I showed you, with a very broad range of biological applications and disease models. So this is actually very exciting. But as I said, it often requires a combination of techniques to go from one scale to the other. And what can be done now in correlative microscopy, which is also one of the main missions for us, because this is really something new and hard to find as a service, is this. So this is the current state of the art of correlative light and electron microscopy at cryogenic temperature, where you are able to image your cell of interest, which is growing on the EM grid as the support. You do light microscopy at cryogenic temperature, then you use a FIB milling device, or Foggius ion beam milling, to thin down a central region of the cell to produce this lamella, because otherwise the cell is too thick for transmission electron microscopy. So after thinning that down, you're able to do cryo-electron tomography on that and get ultrastructure level. You can also go back to light microscopy to make sure that your region of interest is actually retained in the cell. This sounds good and easy, would be great, right, to combine function and structure information from the two methods. However, multimodal imaging workflows are very complicated. They require a large number of devices, many steps, and there are so many things that nowadays can go wrong that pose challenges to the actual application. So to address all those problems, like connecting instruments and technologies that have been in the past developed independently from each other, and now they need to meet again in the middle, EMBL is leading a new consortium called Imagine. This is a European Commission-funded project that involves, I think, 17 or 18 partners from all over Europe, including some industry partners, and the aim is to, again, even further increase our ability to study structure and function of biological specimens across the scales. So not only doing light and electron microscopy on those, but we want to combine X-ray imaging to this, and of course our developments in advanced AI image analysis. And all these workflows will, of course, need to be rapidly transferred to the service facilities that are part of the consortium, thanks to the involvement of the research infrastructures like Instruct and Eurobioimaging. So I could talk for days about correlative approaches, but I just want to give you one glimpse of what this can actually mean to go from a full organism down to a molecular machinery. And this is the work of our colleagues at the Max Planck Institute in Martinsried, from the group of Jürgen Plisko, and my colleague Yulia Mahamid at EMBL Heidelberg. And what you see here is a vitrified specimen. It's a 3 millimeter of diameter planchette, which contains worms, C. elegans worms. Of course, you're not able to see the worms because this is just a slab of ice. And in scanning electron microscopy, you only see the surface of the specimen. So how do I find my worms? Where? With CLEM. You correlate SEM data with light microscopy. And by this overlap, now you're able to identify the worm. And if you have a specific region of the worm that you want to investigate with structural biology, you can make a nanobiopsy. And with this kind of very fancy tweezers, you can lift out this chunk of material that can be deposited on a receiving grid. This is still way too thick to be imaged by transmission electron microscopy. So you have to apply focused ion beam milling to really thin down to 200 nanometers. And again, there you see the step and what is the aim? Well, on that very thin lamella, you can do electron tomography and therefore get structural information about, for example, the ribosome and how translation changes in the cells of the C. elegans, maybe under different metabolic conditions or stress conditions. So really going from the full organism and investigating at the molecular level biological processes. So infection biology, right? So we are also here to discuss some of the new transversal themes, and now we transition to maybe problems that are more related to our health. And the question is are infectious diseases still a problem, or this is behind us? I think we all know that we have many outbreaks of new infectious diseases. SARS-CoV-2 is a clear example. Diseases that have been affecting us for many years are still there and kill thousands of people. The antimicrobial resistance will be clearly a problem of the future, which has been clearly recognized by World Health Organization. And even if we have solutions to the infectious disease, we still have to fight against some portion of the population that doesn't want to listen. So yes, infectious diseases need to be tackled, and to do so, we are studying the biology of our pathogens, and mainly we study host pathogen interactions, responses of the host to the pathogen, how to develop new drugs to fight those infections, and platforms, bioinformatic platforms to allow access to all this wealth of data that is produced. So in the infection biology transversal team, the aim is to develop new technologies. I mean, here you see an example from the Maria Bernabeu lab, where microfluidic chips have been designed to study how the Plasmodium, so malaria parasite, can infect blood cells directly within a capillary. I mean, this is really crazy that you are able to reproduce the actual environment of the host and then study at the structural level parasites. I showed you how high-resolution cryo-electron tomography can show you structural information within cells, and in this case we were able to identify how antibiotics bind to the ribosome directly within living cells that were plunged frozen. And of course, all these new technologies and strategies have to be made available, as I explained, by creating data platforms that can be accessed and investigated by everyone in the community. So I think one great example that shows the power of life sciences and molecular biology in tackling infectious diseases is what happened during the SARS-CoV-2 infection. EMBL was very active at all scales to contribute to the knowledge about this new disease. So we have great examples of how CLAM and volume imaging has been used to understand the viral replication. The development of drug and vaccine was also part of our efforts through the collaboration with BioNTech that used EMBL's small angle X-ray scattering beamlines. And then, of course, all the studies about the epidemiology and the creation of the COVID-19 data portal for global sharing of the data we've been creating. So the infection biology theme involves many groups at EMBL that are scattered all over our sites, which each of them has their unique capabilities of technology and instrumentation. you see that the four leading labs that coordinate those efforts are in the four different sites: Eva Kowalinski in Grenoble, Maria Bernabeu in Barcelona, Jan Kosiski in Hamburg, and Nassus Tipos in Heidelberg. And here is a summary of of some of the activities that the transversal team is doing. We have synergy grants and common retreats, and this is again an opportunity for you to contact us and collaborate on specific topic of interest. And we have a regular meeting both to understanding which direction EMBL should move to fight infectious diseases, and we continue our effort on SARS-CoV-2 work, and basically building also the new level and the next generation of infection biology research, and this is why we are scouting for new talents. So if time allows, I'm not sure, should we stop here, or otherwise I can give you... I just thought I can show you what can we do, right, with imaging, right? So this is very specific. This is maybe not really related to the new activities of infectious biologists, but I just wanted to show you briefly what cryo-EM can do for infectious diseases. So here, this is HIV. I think we are all familiar with this virus, unfortunately. And at the structural level, we have the structural protein, polyprotein GAG, which assembles underneath the membrane of an infected cell. And this budding particle at some point will bud as an immature virus. This is not infectious. When the protease, the viral protease is activated, we have a dramatic structural rearrangement of the virus. The conical core is formed and it encases the genome. This virion now is infectious, can go to the next cell, contact the cellular receptor, and release the cone inside the new cell. So it's clear that this process is where infectivity happens, and therefore studying this transient state will allow us to understand how to tackle the HIV. Cryo-T can come in handy because we really have the structural information about both the immature and mature virions. But what is cryo-T? I mentioned it a few times, but I've never shown you what it really means. So we use transmission electron microscopy to get images of the virions that have been purified and frozen in a thin layer of ice. Then we need to tilt the sample within the microscope and acquire a series of images. Why? Because those viruses are all different from each other. So we cannot get any structural information from one image. We need multiple views. And if you have a unique object, the only thing you can do is to tilt that object within the microscope and acquire a tilt series. And this is what you see now. So this is a collection of, in this case, 41 images at different angles of the same virus. I think, I mean, the projector doesn't help, but I can tell you there is not much to learn from that. You really need to use a computational approach to reconstruct the tomogram. The tomogram is a volume that represents the Coulomb potential, let's simplify it, the electron density of your specimen. And now we are slicing through the volume, and you can appreciate the ultrastructure of the virus. Those are enveloped, so you see this spherical membrane that encloses the capsids. The capsids are in the mature form, so they have these cones that encase this black blob, which is the viral genome. Great, ultrastructure information is very useful, but what if we want to know at the structural level how those viruses are really formed? Well, we apply a technique called subtomogram averaging. So you identify the repeating unit along the feature of interest. In this case, we know that the capsid protein forms hexamers to form these cones. And by rendering the cones, we can then understand how the individual hexameric units are arranged to form those cones. So from just the ultrastructural overview of the viruses, we can then go to the secondary structure level and get a structure of the subunits, hexamers and pentamers, and how they assemble to form a full intact core of the infectious virus. And you see, based on the position of the pentamers that are depicted in blue, HIV can have all sorts of shapes of its capsid, usually conical, but not always. And then the next step is, okay, now I know the structure of the full native core that has not been produced in vitro, but just observed within the virus. But what happens to the cone when it is released into the cell? So there were many ideas, hypotheses on how the genome was protected along the travel from the membrane of the infected cell to the nuclear pore complex, where it has to enter to integrate into the host genome. But the problem was always one. The nuclear pore complex is too small for a HIV capsid to go through. But it was hard to understand how the genome, while being replicated, was protected and therefore not triggering the response of the host cell. So this is the work conducted by Martin Beck. At the time, he was a group leader at EMBL. Now it's a Max Planck director in Frankfurt, and my PhD supervisor, Hans-Georg Kreuzlich from Heidelberg University. So they decided, okay, fine with the hypothesis, just let's see it within the cell. And this is what you see within the cell. Pretty much nothing. I mean, this is very noisy, all gray. And I'm telling you that I see an HIV cone that has traveled along a microtubule, and now it's approaching a nuclear pore complex. I'm not crazy, but this is, why we need light microscopy to really tell us where to look. And now I'm showing you, after you've done CLEM, the result. I mean, you can really render the conical core in purple that traveled along the microtubule in red, and it's really approaching the nuclear pore complex on the nuclear envelope. So those are quite complicated workflows, as you can see here, where you first have to prepare your cells and image them with light microscopy to, well, here it's hard to find, but there is really a very faint signal in this small white box that points to one individual core in the full cell. So you're really looking for the needle in the haystack. But that leads to you seeing for the first time that actually a conical core can indeed go through the nuclear pore. How? I told you that this was too small. Well, it was too small because the nuclear pore complex structure was first solved in vitro, purifying the pore complexes. But actually, the nuclear pore complex has been shown in living cells to change size dramatically from 40 to 60 nanometers, and this depending especially on the metabolic state. So in living cells, the HIV cone can indeed protect the genome while being replicated throughout the journey to the nuclear pore complex, then pass through the pore and release the genome for integration into the host. So I hope that this was a very brief overview, but this is an actual case where both structural biology, application with EM, and light microscopy allowed us to understand the life cycle of quite an important virus. And well, thanks for your attention. I'm very happy to answer any question.